So today we are going to discuss one of the problems which was there in MSCB 2022. MHC, which is the major histocompatibility complex. So MHC1 is expressed by almost all normal cells, but not abnormal and the infected cells. Cells of which of the following lymphoid lineages recognize the tumor cells and the virus infected abnormal cells by the absence of MHC1. So for this we need to know what is the background, how these T cells, B cells, NK cells get activated, and then we ourselves can answer this question, which is not at all tough. So let's brush up. What is immune? There are two types of immunity: innate immunity and the acquired or the adaptive immunity. Innate immunity, which is non-specific, no memory response is needed, it occurs rapidly. Now this neutrophils, macrophages, monocytes, dendritic cells, NK cells, complement system, everything falls under this innate immunity. Acquired immunity or the adaptive immunity. It is highly specific, there is memory response and it develops over longer period of time. The T cells, B cells and the circulating antibodies comes under this adaptive immunity. Now coming to MHC, which is what? Which is the major histocompatibility complex. There are two types of MHC, MHC1 and MHC2. These are encoded by the HLA genes. What is its function? Its function is to present the antigen fragments to the T cell and bind to the T cell receptor. So MHC is actually present on the antigen presenting cells, which presents this antigen to the T cells and it binds to the T cell receptor. Now let's see about how the T cell gets activated, how the B cell gets activated and what is the class switching, and about the NK cell activation. Now coming to the T cell activation. Now we are learning about the T cell activation. This images that I have pasted here, these are from the first aid USMLE step 1 book. Okay, now the T cell activation. T cell activation requires two signals. One is signal 1 and another one is signal 2 or the co-stimulatory signal. When these two signals are present, then only the T cell will get activated. Now see, what are the signals? Exogenous antigens. They binds, see, suppose there is some exogenous antigen, okay? This will be presented to MHC2 in the antigen presenting cell, okay? Then it will bind to the T cell receptor. It binds to the T cell receptor of the CD4 plus, which we call the T helper cells. Always remember one thing, that there is always this sum is actually going to be 8. That means 2. This is MHC2, which when will bind to the CD4 plus T helper cells, it is going to be 8. And if there is endogenous antigen, then it will be presented in MHC1. It will bind to the CD8 plus T helper cell. Okay, so always this multiplication total will be 8. This is nothing but for our remembering purpose. That for the exogenous antigen, it binds to MHC2, activates the CD4 plus T helper cells. And for endogenous antigen, it is expressed through the MHC1, which binds to the CD8 plus cytotoxic T cells. Okay, done. So this is the signal 1. Now what is the signal 2 or the co-stimulatory signal? It is actually the B7 protein. The B7 protein of the antigen presenting cell. Okay, it binds to the CD28 of the T cells and there is the signal 2. When these two signals are there, then there will be T cell activations, there will be cytokines, different type of, and it will kill our infected cells. So this is the activation of the T cell. Now coming to the B cell activation and the class switching. Now see, this is the activated CD4 plus T cell. Okay, then there is a B cell mediated endocytosis. That means this antigen it comes in the B cell, then it is get expressed by the MHC2, which then binds with the T cell receptor of the TH cell. Another thing is that is a CD40 of the B cell. It binds with the CD4L, that is the CD40 ligand of the T cell. Okay, after this what happens? There are several cytokines that is released from our T cells. This can be interleukin 4, interleukin 5. There are the class switching of the B cells and it matures to the plasma cells, which produces the several antibodies which is necessary. So this is the T cell mediated activation of the B cells, which requires this MHC. Right? Now coming to the NK cell activation. NK cell is what? It is actually a type of our lymphocyte. So let us see, this is a diagrammatic representation. This NK cell is having an inhibitory receptor and an activating receptor. Okay. In normal cell, we have this MHC1. MHC1 is expressed in almost all the nucleated cells except RBCs. Okay. So in normal cells, we have this MHC1. This MHC1, it gives an inhibitory signal to the NK cells. See, it binds with the inhibitory receptor and there is no activation and no killing. But in case of tumor cells or the non-self cells, what happens? There is no MHC1. So this gets activated. It releases several perforins granzymes, which causes the apoptosis of the viral infected cells, the tumor cells or the non-self cells. This is one process. When there is an absence of MHC1, then NK cells gets activated. When there is absence of MHC1 in the target cells, then the NK cells gets activated. It releases various perforins, granzymes, and it just kills our Dushman cells. Okay. Now coming to another process, which is the antibody dependent cell mediated cytotoxicity, which we call the ADCC. What happens in non-self cells? There are certain antigens, certain antibodies binds to it, and the CD16 Fc receptor of the NK cell, it recognizes these antibodies, and then there is degranulation and again release of the perforins granzymes, and it kills the infected cells. These are the two processes actually in which the NK cells gets activated. So this is, however, a very vivid and also a crisp idea of how this T cells, B cells gets activated. Now coming to our question once again. This is actually written here. MHC1 is expressed by almost all normal cells, but not the abnormal and the infected cells. We have known about it. Cells of which of the following lymphoid lineages recognize the tumor cells and the virus-infected abnormal cells by the absence of the MHC1? Which one? Now only recently we have learned it is the NK cells, that is the natural killer cells. In the absence of MHC1, there is no inhibitory signal to it, and it kills the target cells. I hope this helped you, and we will be up there with some new videos and new interesting topics. Thank you.